Hello students. So now we will discuss the second video of Puccinia, and in the first video we have studied the two stages of Puccinia, that is the uridial stage and the teleal stage, and in this video we shall study the remaining stages of Puccinia. So this is Rekha working as Associate Professor in Government Science College, Bangalore. So the life cycle of Puccinia graminis, as you all know, it is divided into 5 different stages based on the nature of spores produced. They are the uridial stage, teleal stage, basidial stage, pycnial stage and the aecidial stage. Uridial stage and the teleal stage is passed on the monocot plant, namely the wheat plant, which acts as the primary host. The basidial stage is passed on the soil or on the harvested wheat plant. The pycnial and the aecidial stage is passed on the secondary host, namely the barberry plant. Wheat plant is a monocot plant, barberry plant is a dicot plant. So coming to the third stage, that is the basidial stage. So this is characterized by the formation of basidiospores. So the teletospores, after taking a period of rest, it germinates independently and it produces the basidiospores. So each teletospore is bicelled and binucleate spore. So from each cell of the teletospore, a short hyphae arises. So the two nuclei which are present in the teletospore now fuses to form a diploid nucleus. So this nucleus divides twice, during which meiosis occur, resulting in a row of 4 haploid nuclei. So as they are provided with 2 cells, as each cell divides, totally you have the 8 haploid nuclei. So 4 in one cell and the other 4 in the other cell. They are separated from each other by the transverse walls. So this 4-celled structure, so you can see in the figure D, though you can see from one of the cell of the teletospore a 4-celled structure, and this structure is called as promycelium or basidium, or it is also called as epibasidium. So each of the 4 cells produces a single colorless uninucleate basidiospore on a shorter sterigma. So the fungus is heterothalic, hence the 2 basidiospores belong to plus strain and the other 2 belong to minus strain. So this is from one cell of the teletospore. As 2 cells are involved, totally 8 spores are produced. Out of 8, 4 spores produces plus strain and 4 spores produces the negative strain. So these are discharged, these basidiospores, they are discharged by an explosive mechanism and they are easily dispersed by wind. So these basidiospores germinates only on the barberry plant. So if it falls on the wheat plant, it will not germinate. So further germination of the basidiospore is seen only on the barberry plant. So on the barberry plant, it produces, during germination, it produces a small slender tube. So when it produces a small slender tube, it penetrates into the epidermis directly and branches freely, it forming a haploid monokaryotic intercellular mycelium composed of uninucleate cells. So this stage is called as the pycnidial or spermogonial stage. So the mycelium carries either plus or minus strain, and both plus and minus strain may grow side by side in the same barberry leaf. So the germination of the basidiospore leads to monokaryotic mycelium. So this monokaryotic mycelium branches and grow vigorously, producing dense mats of uninucleate hyphae here and there between the upper epidermis and the palisade tissue of the host leaf. So these are the primordia of the spermagonia, or it is also called as pycnia, which is seen as small yellow or red patches which appear on the upper surface of the infected leaves. In addition to pycnia, the monokaryotic hyphae, it grow towards the lower surface of the barberry leaf and they form the Aecidia primordia just opposite to the primordia of Spermagonia. So, primordia of Spermagonia, it gives rise to a flask shaped structure which is called as Spermagonia or Pycnidia, which may either belong to plus or minus strain depending upon the nature of basidiospores. A mature spermagonium is oval to flask shaped structure which projects from the leaf surface by a pore called the ostiole. So, from the base of the spermagonium, a large number of elongated uninucleate cells called the spermatiophores arises. These spermatiophore, it cuts off uninucleate conidia like bodies which are called as pycnidiospores or spermatia. So, these spermatia are small oval to spherical smooth walled spores. So, you can see the diagram here, the flask shaped fruiting body, and you can see the pycniospores or the spermatia inside the flask shaped fruiting body, and in addition to the paraphysis, that is sterile hyphae, the flexuous hyphae or the receptive hyphae are seen near the ostiole, which acts as the female sex organ. So, the hyphae adjacent to the ostiole develop into periphysis which protrude beyond the ostiole. In addition, several receptive hyphae extend upwards and these are the female gametangia. So, these receptive hyphae project from the spermagonia. Pycnidiospores function as the male cells and bring about the diploidization of the monokaryotic mycelium. They carry the same plus or minus strain depending upon the mycelium which produce the spermogonium. So, these spermatia, they are exuded out in small droplets of nectar from the ostiole. The insects, they carry the spermatia, so they are attracted to the nectar, so they come and suck this nectar. So, during their feeding, so these spermatia, they get attached either to the mouth parts or the leg parts of the insect, and when they visit another pycnidium, thus they are carried from one pycnidium to the other pycnidium. So, if plus strain of the pycniospore comes in contact with the receptive hypha of the opposite strain, they fuse and the nucleus of the pycnidiospores passes into the receptive hyphae and moves downward, establishing a dikaryon condition. So, you can see the diagram here with the spermatia and the receptive hyphae. The nuclei of the spermatia, it passes through the receptive hyphae and makes the receptive hyphae dikaryotic. So, the Aecidial primordia, which are initially monokaryotic, do not develop until they become dikaryotic. So, the next stage, the last stage is the Aecidial stage. So, fusion of the spermatium with the receptive hypha is followed by the migration of the spermatial nuclei into the receptive hypha through the septal perforations of the mycelium to the basal cells of the Aecidial primordia, which thus they become dicaryotized. If there is no dicaryotization, the protoaecidium aborts and the Aecidial cup is not formed. So, the dicaryotized basal cells, they are commonly known as acidiospore mother cells or sporophores. So, the edge of the peridium forms the wall of the Aecidial cup. It bends upwards and it forms a bell shaped structure. So, you can see the bell shaped structure of the Aecidium. So, from these, in this diagram, you can see a chain of binucleospores emerging out, and these are the acidiospores which are cut off and alternating with the disjunctar cells or the sterile cells. Simultaneously, the neighboring marginal cells from the peridium which surrounds the cells in the Aecidium. The Aecidial cup remains partly embedded in the leaf tissue and it partly projects above it. So, you can see this diagram also with the pycniospores, the pycnidium just above the upper surface, and the Aecidium in the lower surface. So, these Aecidial cup appears on the lower surface of the infected leaf as white cups filled with the golden yellow powder of the spores. So, the developing Aecidium exerts pressure on the leaf tissue and ruptures the peridium and the lower epidermis, and as a result the exposed binucleate Acidiospores fall from the Aecidium. The Acidiospores are unicellular, binucleate, thin-walled and polyhedral in shape. The Acidiospores, they are unable to reinfect the barberry plant, but they can infect only the wheat plant, thus repeating the life cycle. So, thus in the life cycle of Puccinia graminis, five different types of spores are produced. The Spermogonium, also called as the Pycnidium, bearing the spermatia and the receptive hyphae, Aecium bearing the Acidiospores, Uradium bearing the Iridospores, Telium bearing the Teletospores, Basidium bearing the Basidiospores. So, to summarize the life cycle of Puccinia, so on the wheat plant, the first Acidiospores germinate, later dikaryotic hyphae form the Uridia bearing the Iridospores. So, as the season changes in the late summer, it produces another type of spores which is called as Teletospores inside a pistule which is called as Telia or Teletosaurus, and again the Teletospores produces the Basidiospores on the Basidium, and on the barberry plant these Basidiospores germinate and produces the Spermagonia or Pycnia bearing the spermatium and the receptive hypha. The nuclei of the opposite strain remain in common protoplasm without the fusion, forming the dikaryon. Then again in the same barberry plant, Aecidial cups are produced, which produces the Acidiospores on the lower surface of the barberry leaf, and these Acidiospores again they reinfect the wheat plant. So, this is the diagrammatic life cycle of Puccinia graminis, and this is the life cycle of Puccinia. So, the wheat plant produces Iridospores initially, and these Iridospores it keeps on increasing the Iridospores, and it gets infected from, it causes infection from one plant to another plant till the whole crop is infected. And the second type of spores is the Teletospores. In the Teletospores, the fusion of the nuclei takes place, resulting in the formation of diploid nucleus. It undergoes meiosis. So, it results in the formation of the Basidium. Basidium produces two kinds of Basidiospores, two of them belong to plus strain and the other two belong to minus strain. So, these Basidiospores they infect the secondary host named the barberry plant. So, on the barberry plant it produces the Pycnidium and the receptive hyphae, and Pycnidium produces the spermatia. The spermatia and the receptive hyphae they fuse with one another, forming the spermatization, and the spermatization leads to the dicaryotization of the Aecial cup which is present at the base of the leaf. So, these Aecium it produces the Acidiospores. So, these Acidiospores again infect the wheat plant. So, this is the graphic life cycle of Puccinia graminis. So, to summarize the life cycle of Puccinia, so Puccinia causes black stem rust on the wheat plant, but the stem rust, the rust disease has been seen in most of the cereal crops like wheat, barley, oat and rye, but the typical black stem rust is caused by Puccinia graminis in the wheat plant. It is an heterogeneous, Puccinia graminis is a heterogeneous, polymorphic, macrocyclic, obligate fungi, and the asexual reproduction begins on the graminaceous hosts, and sexual reproduction begins in the resting spore stage, and culminates on the alternate host. Thank you.